Let us know about the process of Kelvin's cycle. It is also known as light independent phase. In the year of 1956, the scientists Benson and Kelvin first introduced the process of Kelvin cycle in chlorella sp. So that's why it is also known as Kelvin cycle. And the Kelvin cycle is divided into four phases. That is, first is carboxylations, then reduction, then synthesis and regeneration. Now come to know the carboxylations process. So at first the RUBP, that is the ribulose bisphosphate, with the help of carbon dioxide which is present in the air and with the help of the enzyme, that is Rubisco. So RUBP, with the help of carbon dioxide and with the help of the enzyme Rubisco, it will form 2-carboxy 3-keto-arbinitol phosphate, that is also known as DPOB. So DPOB means that 2-carboxy 3-keto-arbinitol 1,5-bisphosphate, and it is an unstable compound. That's why it also dissociates into two molecules of compound, permanent compound, that is phosphoglyceric acid. So in the first step of carboxylations, that is the RUBP, which is present in the cell, it attached with the CO2, and with the help of the enzyme Rubisco, it will form 2-carboxy 3-keto-arbinitol 1,5-bisphosphate, which is unstable, and it will dissociate into 3-PGA, that is 3-phosphoglyceric acid. And next step is reduction phase. So in this phase, the 3-PGA, which is formed with the help of ATP and also the enzyme phosphoglycerokinase, with the help of the enzyme phosphoglycerokinase, it will form 1,3-bisphosphoglyceric acid, that is 1,3-BPGA. It is the short form of bisphosphoglyceric acid, and also the ATP will convert into ADP. So in this, the reduction phase, the 3-PGA, which is formed in the carboxylation phase, it will form the 1,3-BPGA, that is 1,3-bisphosphoglyceric acid. And in the next step, the 1,3-bisphosphoglyceric acid will, with the help of NADPH and also hydrogen compound and the enzyme, that is glyceraldehyde 3-phosphate dehydrogenase. So this is the enzyme, that is glyceraldehyde 3-phosphate dehydrogenase. It will form the 3-phosphoglyceraldehyde, that is 3-PGALD, and the NADPH and H plus also form the NADP plus inorganic phosphate. So the 1,3-bisphosphoglyceric acid, with the help of NADPH and H plus and with the help of the enzyme, that is glyceraldehyde 3-phosphate dehydrogenase, it will form the 3-phosphoglyceraldehyde. And in the up next, it will enter into the synthesis phase. So next, in the synthesis phase, the 3-PGALD with the help of another enzyme, that is triose phosphate isomerase. It will form 1,3-dihydroxyacetone phosphate, that is DHAP. So the 3-PGLD, which is formed in the reduction phase, it will enter into the synthesis phase, and with the help of the enzyme, that is triose phosphate isomerase, it will form 1,3-dihydroxyacetone phosphate. And the 3-PGLD also act with the DHAP. 3-PGALD also act with 1,3-dihydroxyacetone phosphate, and with the help of the enzyme aldolase, it will form fructose 1,6-bisphosphate. Clear? The 3-PGLD and is react with 1,3-dihydroxyacetone phosphate, and with the enzyme aldolase, it will form fructose 1,6-bisphosphate. And next, the fructose 1,6-bisphosphate will react with H2O, that is water, and there is another enzyme, that is fructose 1,6-bisphosphatase enzyme. So the fructose 1,6-bisphosphate with the help of H2O and fructose 1,6-bisphosphatase, it will converted into fructose 6-phosphate. Fructose 6-phosphate. And also the inorganic phosphate. Clear? And the fructose 6-phosphate with the help of another enzyme, that is phosphohexoisomerase. It will form glucose 6-phosphate. And the glucose 6-phosphate with the help of ADP and also the phosphatase enzyme, it will form glucose, and also the ADP will be converted into ATP. Clear? So the synthesis phase, first is the 3-PGLD with the help of triose phosphate isomerase enzyme, it will form 1,3-dihydroxyacetone phosphate. And in the next stage, the 3-PGLD, that is phosphoglyceraldehyde, and with the 1,3-dihydroxyacetone phosphate, and with the help of the enzyme aldolase, it will form fructose 1,6-bisphosphate. And in the next step, the fructose 1,6-bisphosphate with the help of the enzyme fructose 1,6-bisphosphatase, it will form fructose 6-phosphate. And the fructose 6-phosphate with the help of the enzyme phosphohexoisomerase, it will form glucose 6-phosphate, and the glucose 6-phosphate with the help of the enzyme phosphatase, it will form glucose plus ATP. I hope it will be clear for you. Up next, there is a phase called regeneration phase, where the fructose 6-phosphate, that is the fructose 6-phosphate with the help of the 3-phosphoglyceraldehyde and the enzyme that is called transketolase, it will form the erythrose 4-phosphate, which is a 4-carbon compound, and also another compound, that is 5-carbon compound, that is xylulose 5-phosphate. So in the next is the erythrose 4-phosphate, it will act with dihydroxyacetone phosphate, that is DHAP, which is a 3-carbon compound, and with the enzyme aldolase. Here the enzyme which is present, that is aldolase. It will be form the pseudoheptulose 1,7 bisphosphate. Pseudo So the erythrose 4-phosphate, with the help of the enzyme aldolase and with the help of a compound, that is 3-carbon compound, that is DHAP, it will form the pseudoheptulose 1,7 bisphosphate. And in the next step, the pseudoheptulose with the H2O will be attached, and the enzyme that is present is pseudoheptulose 1,7 bisphosphatase. It will be formed that pseudoheptulose 7-phosphate. Pseudoheptulose 7-phosphate, and also the inorganic phosphate is occur. And in the next step, the pseudoheptulose 7-phosphate with the help of PGALD, that is phosphoglyceraldehyde, and with the help of another enzyme, that is transketolase, it will be formed ribose 5-phosphate. It will form ribose 5-phosphate, and also another compound which is also a 5-carbon compound, that is xylulose 5-phosphate. Two compound always 5-carbon compound, and the ribose 5-phosphate in the next step with the help of isomerase enzyme, it will be formed ribulose bisphosphate 5-phosphate. And xylulose 5-phosphate also, here is act another enzyme, that is epimerase. It will also form ribulose 5-phosphate, and the ribulose 5-phosphate with the help of ATP and also phosphorylose bulose kinase. Kinase, which is an enzyme, it will be formed ribulose 1,5 bisphosphate. And also ADP will be formed. I hope it will be helpful for you, though it is a very big process, but it is very important topic, and I hope you will get it easily. Thank you so much for watching my video. Please like, share and subscribe my video. Thank you so much.